Welcome to the MIST Help video. This video demonstrates how to use the Molecular Interaction Search Tool, or MIST. The MIST tool is a comprehensive source of gene or protein interactions. At MIST, you can query known physical interactions as well as infer interactions for genes that are of interest to you. So, the MIST tool, how do you search the MIST tool? So, the first step is to choose a model organism or species. MIST supports 9 species, so you just simply select the tab at the top corresponding to your species of interest. The next step is to select the search type. There are two options. The protein list option is enter a list of gene or protein identifiers. This type of search will return all interactions relevant to your input genes. For the protein list option, there's a drop-down menu, and you can choose either to find all interactors or for your input genes or just interactions within the input gene list that you've entered. The second option on search is protein pairs. If there's an interaction annotated in MIST for this pair, then the interaction will be displayed. The third step is to enter your list of genes or proteins of interest. There are two ways to do that. You can either enter them into this big text box, separated by a comma, space, or new line, or you can choose to upload them from a file. The file can be .txt or .csv format. Supported identifiers include Entrez gene ID, gene IDs, gene symbols, protein symbols, fly-base IDs, and worm-base IDs. So we'll enter a couple of gene symbols into the search box. Next step is to choose a network type to query. Some of the available options are physical protein-protein interactions, interlogs, which are inferred physical interactions supported by the data from different species, predicted kinase substrate interactions, predicted domain-domain interactions, genetic interactions, correlation networks based on phenotype, phosphorylated sites, or gene expressions. So under protein and protein interactions, you can be more specific. You can click to see more options. You're able to specify assay type and or studies from a pop-up list. The menu contains information from the top high or medium throughput studies as defined by the number of protein-to-protein interactions published in the study. In this case, I'll just select all. When you're ready to search, click Submit. You'll be directed toward this is the results page. The results page is broken up into two sections. On the top is a network display, and the bottom is a network that contains data tables. So let's take a look at the network display. It shows the interactions as edges or lines, and proteins as nodes. Different color edges indicate different types of interactions, and you can see a key to that on the upper right-hand side of the screen. If you want to expand your network from a specific node, you can simply double-click on it and the additional interactors will be displayed. To go back to the original one, simply click Submit. If you want to add or remove a type of interaction using this checkboxes on the upper right-hand side, simply check the ones you want and click Submit, and it will repeat your search. And update the table at the bottom. So going back to the top, some searches, such as interlogs, have filters. An interlog pair inferred from just one species will have a score of 1. An interlog pair inferred from all available species in MIST will have 9. The filter lets you change the cutoff. such as only to view the pairs inferred based on the evidence from 2 or more species. The next option is the change layout option right here. From here you can change the layout of your graph. Circle, concentric, grid, and random. Let's look at our network tables. The network tables display all the protein pairs in the graph above. There's a drop-down menu, lets you select how many to see. If multiple types of interactions are selected, you'll see a separate table for each interaction displayed below here in this area. If no interactions are found for a subset of the protein or protein pairs entered, the inputs will be shown under a no interactions table in this area. This is a demonstration of enrichment analysis using MIST. So again, choose the species you like to work with, find interactors with an input, and clicking the example button fills in a bunch of example genes. Network type you like to select and hit submit. If you've done a search using the Find Interactors with an Input option, you can also do an enrichment analysis. To choose the option, look for the big red button on the right-hand side of the screen, just below the Change Layout drop-down. Clicking the Enrichment Analysis button will open a new page. So this is the enrichment analysis. MIST calculates an enrichment score based on the number of interactions among your input proteins as compared to what would be expected at random. Sometimes it takes a little while to load, but the display will be a histogram showing the random distribution in blue and your interactions in orange. Below this you can see a table that contains a number of interactions, p-values, mean, and standard deviations. The tooltip, by hovering over the graph, shows the exact number of values. Let's go back to the main search page and talk about downloading your results. So to download your graph network diagram, you simply right-click on the graph and select the options for downloading. You can download it as PNG, JPEG, or Cytoscape JSON file. And that's it. Looking at the tables, to download an individual table, simply click the Export Table button at the top of each table. And that's it. It will download as a CSV file. To download all the tables at once, you simply click the Export All Tables button. And it will download all the tables, again, in Excel format. Some statistics about MIST. This page contains a summary of the interactions and proteins in our database. For more details on our enrichment analysis calculations and other queries, you can visit the About Us help page. And there's more information here about MIST. And feel free to contact us using the information in the About Help page. Thank you for watching the MIST introduction video.